Hello, my name is Tom Pollard. I'm a professor at Yale University, and I'm giving a two-part talk about how actin polymerization produces force to allow cells to move, and also for other processes such as actin-mediated endocytosis. Here on this slide, you can see the dendritic nucleation hypothesis, which we put forward in the late 1990s, and were summarized in this beautiful diagram by Dyke Mullins in 1998. As you heard in the first half of this talk, this is a summary of the biochemistry that we understood at that time. But you might want to ask whether this nice biochemistry is actually what happens in live cells. The problem was to find the right biological setting in which to ask and answer this question. At the leading edge of a motile cell, like this keratocyte at the top, the actin filaments, shown here in this micrograph, are so tightly packed, and the events are happening so rapidly that we don't have any temporal or spatial resolution. All the reactions are superimposed on top of each other in time and space. However, there are examples where these same proteins are used for other cellular functions and where we have better temporal and spatial resolution. For example, here we have a Fission yeast cell. And the bright little spots, which you can see here in this fluorescence micrograph, are sites of actin-mediated endocytosis. Over on the left, you can see electron micrographs of thin sections from the laboratory of Pietro di Camilli, showing a coated pit, an invagination of the plasma membrane, and a little vesicle that's pinched off from the plasma membrane during the process of endocytosis. And it turns out that the proteins used for the motility of this animal cell and endocytosis in the yeast cells are exactly the same. So we can use the yeast cells as a test bed for our ideas. Now these assemblies of actin, which are outlined here in this with a circle, are very small compared to the leading edge. So that's how big one of these things would be in the yeast cell. And a comparable sized area would only be about this big at the leading edge of a motile cell down here. So we're going to be looking in the next few slides at a tiny little microcosm of what happens at the leading edge of a cell. Over here on the right, you can see the first frame of a time-lapse movie made by Volodya Sorokin. And let me point out down here at the bottom 3 bright spots. These are sites of actin-mediated endocytosis, and some of them are red, and some of them are yellow, and some of them are green, like this one right over here. Those are just different points in time for a process that it's very much the same at every one of these sites. So now I'll play the movie, and you can watch these sites down here at the bottom end of the cell. And what you'll see if you look closely is that they start out red, they turn yellow, and then they turn green. Well, the red signal is coming from a nucleation promoting factor called WASP, and the green is coming from the ARP2/3 complex. When both of them are together, then it's yellow. So here are frames from a movie like that. Up at the top, you can see time in seconds, and you can see our little spot, which we call an actin patch, starts out as being red over here on the left. In the middle, it's red and green, so it's yellow. And eventually, the red and green separate from each other as both of them fade away. So the big advantage here is that the action is limited to a very small space, and it's a one-off reaction where the assembly and disassembly take place in a predictable fashion. The timing is consistent with the dendritic nucleation hypothesis. For example, the first frames over here correspond to the recruitment of a nucleation promoting factor, and then the recruitment of ARP2/3 complex, and then the formation of a branch, and eventually the dissociation of both the ARP2/3 complex and the nucleation promoting factor. So the time course of these events in the yeast are consistent with the hypothesis that we put forward for cell motility. Now we could do a good bit better than that by actually quantitating the data in these micrographs, and this is possible due to the work of Zhenshu Wu, a paper in 2005. what Zhen Chu discovered was that you could collect all the fluorescence from a cell. For example, here's some yeast cells, some more yeast cells down here at the bottom, that are expressing two different proteins that are tagged with a fluorescent protein called YFP. So he could count up the total amount of fluorescence in each one of these cells by taking a Z-stack of the cells and adding all the fluorescence together. He then took samples of cells that were expressing tagged proteins at different levels naturally in the cell. And for example, this one is a septin tagged with YFP up here at the top. He ran on a gel duplicate samples of two different loadings of a known number of cells and compared on this immunoblot the intensity of these bands with the intensity of bands of a known amount of purified YFP, which we're using as an epitope tag in this experiment, in addition to giving the fluorescence. And what Zhen Chu discovered down here at the bottom was that the fluorescence intensity is directly proportional to the numbers of the YFP proteins in the cell. So that means that this microscope is a fantastic spectrophotometer, and over 3 orders of magnitude, you get a linear response between the numbers of fluorescent molecules in a pixel and the fluorescence intensity. So that means we can go back to our fluorescence micrographs and turn all those beautiful images into numbers. And so this is an example showing the time course here in seconds of the accumulation and disappearance of proteins at sites of clathrin-mediated endocytosis. So early in the event, you can see over here on the left that the only protein we detect is clathrin, which is a protein that makes an icosahedron around on the surface of the membrane. And there's actually just enough clathrin here to make half of an icosahedron, or a hemisphere. After a little time has gone by, these blue proteins show up and accumulate, first binding one-to-one with the clathrin. After a bit more time goes by, these red proteins show up. Those are the nucleation promoting factors, followed in black by the ARP2/3 complex, and then the assembly of actin. Now, only 5% of the actin is labeled, so the real actin peak, or 6% in this experiment, so the real actin peak is about 20 times higher than this, way up above the screen. So what this shows is that each one of these proteins shows up and disappears over a very predictable time course. And at time 0 on this time scale, the actin patch starts to move away from the plasma membrane. That's how we synchronize them all in time. So with data like this, it puts great constraints on the underlying biochemical reactions, allowing one to propose and test a specific hypothesis for the process. So our hypothesis was the dendritic nucleation mechanism, which you can see up here at the top. Julian Barrow made a mathematical model where he used ordinary differential equations to describe each one of the reactions in this scheme. We assumed that the accumulation of WASP, shown in blue over here, was going to drive the entire process. Now, how WASP gets there is actually an interesting question, but not part of this model. We're just going to let the WASP accumulation drive the rest of the reactions. We assumed the rate constants we'd measured in dilute solution with purified proteins, mostly from the Fission yeast. We assumed bulk protein concentrations that we can measure in the cells with the fluorescence microscopy method I just described. And we can assume that the reactions occur in a little reactor, about 300 nanometers in diameter, and that the molecules can move in and out of this little reactor. And then we can run simulations of the reactions to determine the number of molecules inside the reactor over time. Now, this actually works remarkably well. Over here on the left, you can see a graph showing the time course of the accumulation of molecules in the, at an actin patch. The symbols, like these triangles here, show the experimental data, and the smooth lines show the simulations. So the whole process is robust in the sense that you can vary the assumptions quite a bit, and you still get assembly and disassembly of actin. However, as you will notice in this diagram, that the timing and the amplitude of the accumulation of actin is different from the experimental measurements. So now we can actually learn something about what's going on in the cell by adjusting the parameters to fit the experimental data, and as you see here, and then discover what's different about reality than our model. So in this simulation here, you can see the smooth line fits the experimental data for actin and ARP2/3 complex and capping protein beautifully. And it turns out that a very limited set of parameters will actually allow the simulations to fit the data so well. So you're going to ask, well, did we just make up some parameters, or did these parameters make sense? And it turns out they make absolutely beautiful sense. So here are the adjustments we had to make in the original assumptions. First of all, we had to assume that capping protein binds to the barbed ends of the actin filaments 10 times faster inside the cell than it does in a dilute solution in a test tube. This was actually very good news because we've been interested for a long time in something called an excluded volume effect. In a crowded environment with a lot of inert things like proteins and ribosomes, the molecules are actually crowded together and have higher chemical activities than they do in dilute solution. This is actually a bigger excluded volume effect than we expected, but definitely in the direction that we anticipated. So we expect that many of the reactions inside the cell are going to be much faster than they are in our typical biochemistry experiments. Second, to get the simulations to work, we actually had to assume that the ATP is hydrolyzed and the phosphate dissociates 50 times faster inside the cell than it did in our prior experiments, which were actually done with muscle actin. Now, muscle actin is a very good substitute for cytoplasmic actin for many things, but not for this. It turns out that when Jeff T finally purified the actin from the fission yeast, that it actually hydrolyzes its ATP and dissociates phosphate 100 times faster than muscle actin. So the cellular measurements and mathematical modeling actually got the answer before the biochemists in that case. Third adjustment was we had to find adjustments to form the branches much faster than they did in the test tube, about 400 times faster. Well, one factor is surely an excluded volume effect. Another factor is that we were missing a protein in our biochemistry experiments that speeds up the reactions by about a factor of 5. And so we are actually within about a factor of 20 of explaining this reaction. And the fact that the reactions are occurring on a flat surface, on a membrane, is another factor that could speed up that reaction. Finally, there was no way that the actin could disappear from these actin patches by simply dissociating from the pointed ends of the filaments. This reaction 13 is thousands of times slower than necessary to account for this. So clearly, the disappearance of the actin was not due to depolymerization from pointed ends. And it turned out that the answer was in Dyke-Mullen's original drawing, and that's shown here. That is that the filaments get chopped up by cofilin and diffuse out of this little area where we're making the measurements, and they depolymerize someplace else. So this last result suggested that cofilin had a very important role in taking apart these actin filaments at sites of endocytosis. And so Chen, shown down here at the bottom, tested this by making mutations in cofilin that slowed down the rate of severing quite a bit, by more than 50-fold. And when you replace the wild-type filament, cofilin, with the mutant cofilin over here on the right, he observed that the actin patches were much more sluggish and much brighter when looking for actin here, stained with a protein called GFP fibrin, which was certainly consistent with our idea that cofilin had an important role in disassembling the patches. So as expected, the cofilin has an important role in the turnover of the actin. But he learned something in addition. He got a big bonus from this experiment, as shown here in these two panels. These are quantitative measurements of the numbers of molecules per patch over time. The blue data is from a wild-type cell, and you can see the accumulation and disappearance of actin over 20 seconds, as I've told you about before. That's in a wild-type cell. In a mutant cell with this slowly severing cofilin, the actin accumulates to much higher levels and disappears over a much longer period of time, as we expected for the important role of cofilin. But here's the big surprise at the bottom, and that is looking at one of the adapter proteins that's involved in the early stages of this process. Chen discovered that the adapter proteins accumulate over a much longer period of time in the mutant cells than the wild-type cells. And so that shows that there's some sort of completely unanticipated feedback loop in the system that we hadn't appreciated before his work. This is a diagram showing our idea about what's going on. And the idea is that cofilin severs the actin filaments at these sites of endocytosis, and that the filaments aren't simply disappearing, but they're actually doing something important to make the whole process work properly. So the first step is to sever the filaments and create these small fragments, which are shown here in green. These fragments then diffuse around in the cytoplasm of the cell. We can't actually see them because they're very short, and on average they have one fluorescent actin in them, so they'll look just like an actin monomer in the light micrograph. Up at the top, we see a forming site of clathrin-mediated endocytosis, and the black and purple molecules are the adapter proteins. Those two adapter proteins actually have binding sites for actin filaments, and those two actin binding sites must be active in order for the whole process to work. So our interpretation of this is that the actin filaments bind to these adapter proteins, and they become the mother filaments that get the whole process of actin filament branching started at the site of endocytosis. So there's my summary of what we've learned about the machinery that produces the forces that help cells move in motile cells, and also provides force to invaginate the membrane at sites of endocytosis. Here's Dyke Mullen's beautiful diagram. And I want to emphasize that this is not the whole story. This process at the top produces the force on the membrane that causes the cell to move, but the actual movement of the whole cell depends on other reactions, such as the cross-linking of the actin filaments, adhesion of the cell to the substrate, and the signaling system, which I started with up at the top here, to guide the cell. In the process of doing this work, I think we've learned some things that might be useful to people doing work on other biological problems. So let me just summarize some of the lessons that we've learned from this work. The first at the top here is to pick a good model system to do the studies. We actually did the initial studies on amoebas. Hardly anybody works on amoebas, but they were a fantastic source of actin. That's where ARP2/3 complex was discovered. That's where capping protein was discovered. But eventually we had to turn to a genetic model organism, namely Fission yeast, so we could do the mechanistic studies, which I just described. Secondly, every one of the proteins we've studied has a complicated mechanism of action. Even something as apparently trivial as profilin, which binds actin monomers, actually has a complicated mechanism of action. It took us over 25 years to actually get to the bottom of how that protein works. And those details are necessary to understand the system and to make good models. Connecting the biochemistry to activities inside living cells requires the ability to make quantitative measurements in live cells, so you can collect the data to road test your models. And I've described a method of using quantitative microscopy to do that. Now this process, and virtually all cellular processes that rely on more than a couple of molecules, are too complicated to understand intuitively. You simply can't get your arms around things firmly enough to know whether you're on the right track or not. So the only option that we have available is to make a commitment to a certain hypothesis in the form of a mathematical model, run simulations of that model, and compare those simulations with quantitative experimental data. And then and only then are you able to disprove your hypothesis, and I showed you an example of that, and to make progress as you iterate around between the experiments and the modeling to get closer and closer to the truth. And finally, I hope you noticed that we have taken advantage of time in almost all of our experiments. And I recommend highly to you that you have time on your x-axis in your experiments, because this gives you an extra handle on the system you simply cannot benefit from unless you've carefully engineered that into your experiments. So thank you for listening. I hope you enjoyed finding out about the mechanism of cell motility, and hopefully some of this advice will be useful to you in your work.